Okay.
Well, I'm pleased to introduce our guest speaker today for Denison. Dr. Denison is an adjunct professor on ecology evolution since 2005 at University of Minnesota. Before that, he was professor at UC Davis for 12 years, where he taught crop ecology, and he also spent 7 years conducting research with the USDA. Ford studied ecology at Evergreen State College and earned a PhD in crop science at Cornell University, and was a postdoctoral fellow at UC Davis and at UCLA. Much of Ford's research has focused on studying symbiosis between legumes and the root nodule bacteria rhizobia, with an interesting evolutionary focus, and his work on this topic has been published in journals from Nature to Field Crop Research. He's well known as the author of the book Darwinian Agriculture, that provides a unique perspective on the trade-offs driven by evolution that might provide opportunities for improving agricultural systems, and points out some of the limitations in agricultural biotechnology and of agricultural systems that try to mimic natural systems. And Ford has been invited as a speaker to talk on ideas on his book throughout the US and in 10 or more foreign countries. Something that many in our department will find interesting too. While at UC Davis, he directed their long-term research on agricultural systems, evaluating long-term effects of conventional and alternative agricultural systems. So there are many topics that we would love to hear Dr. Denison talk about, but we are also very excited that today he will be sharing with us some of his research on nitrogen fixation. So we are honored to have Dr. Denison as a speaker today. Thank you and welcome.
Well, thank you. Somehow I got invited to give a couple of talks at the agronomy meetings, virtual meetings coming up in November. One on perennial grains, which I don't know anything about, and one on yield gaps, which I don't know anything about. But today I'll talk about something I do actually know something about. So it's nice to get an invitation closer to my expertise. And I'll go ahead and share my screen. Let me know if it's misbehaving. How's that? Is that okay?
Yes.
Okay. So title of my talk is Cooperation and Conflict in Legume-rhizobia Symbiosis with Implications for Agriculture. And I actually, in this first slide, I'm actually showing here on the left my thesis research at Cornell on nitrogen fixation. The top middle, much of the work I did with USDA was also on nitrogen fixation. This is a device that we invented as part of that work. On the right is gas exchange work that I did some at Davis and some here at Minnesota. And for the last few months, I've been doing much of my research in my basement, and that's the bottom slide. So here's a summary and a little plug for my book. Rhizobia strains differ in net benefits to legumes, and that's partly due to differences in diversion of plant carbon to rhizobial resource hoarding. One of the ways that legumes may deal with this is to manipulate rhizobia, making them fix nitrogen more efficiently. But the one way we know that does this actually blocks the reproduction of the rhizobia. Another legume strategy is to actually impose sanctions, shutting down nodules that are not providing as much benefit. And that's kind of the focus of my current research, is to look to see whether legume cultivars, specifically soybeans, may differ in that trait, and what the benefits could be, benefits and costs to the current year's soybean yield, and possible benefits to future soybean yields in the same field, because we'd like to have a soybean that would only allow the very best local rhizobia to reproduce in its nodules and only release those into the soil for availability of future crops. So here's just a little bit of background. The key reaction that the bacteria carry out inside the nodules is here: nitrogen gas, inexhaustible supply. They consume large amounts of ATP. They produce ammonia, which the plant takes up and uses to make protein. And an important point is that hydrogen gas is released as a byproduct of this reaction. And that turns out to be useful because it's easier to measure hydrogen appearance than it is to measure the disappearance of nitrogen gas. So from the plant's point of view, what the rhizobia should do is take the carbon, take oxygen from the soil atmosphere, respire all that carbon to CO2, use that to make ATP, use that to power this reaction of nitrogen to ammonia. But if we look at real soybean, real rhizobium cultivars, varieties, strains, real rhizobium strains, we find that they actually vary a lot in the amount of benefit that they provide. And this graph is a graph of benefit in the vertical axis. Anything that's not above this line, this is the yield without any rhizobia. So you can see these guys down here really aren't providing any benefit to the plant, but they're also not costing the plant anything, because this axis here is nodule mass per plant. So you can see the reason these don't provide much benefit is they don't make very many nodules, and that means they leave opportunities for better strains to nodulate. What I'm most worried about is strains like this that have a relatively high cost, occupying lots of nodules that could have been occupied by better strains, and yet providing very little benefit. Not zero, but not much. Up here is USDA 110, which is a widely used inoculum strain of rhizobia, and you can see that it has both a lower cost and perhaps because of that lower cost, a substantially higher net benefit. And then most of the strains that are out there are, I don't know, maybe half as good as USDA 110. And that's the problem that I'm hoping to help solve. Now, there are many reasons why rhizobia might be less beneficial. They might just be defective in ways that hurt them just as much as it hurts the plant. But we also know that rhizobia divert some fraction of the carbon they get from the plant into their own reproduction and also into hoarding of this lipid, polyhydroxybutyrate, or PHB. And that obviously comes at the expense of carbon that could have been respired to make ATP and power nitrogen fixation. But how big an effect is that? Well, let's start with the rhizobium point of view. Oh, first, this slide showing there really is a trade-off. Here on the left is, this is actually USDA 110 that I mentioned. This little envelope here has 3 rhizobium cells. This is a cross section, just a bit of a plant cell here with lots of these little envelopes. And you can see these little white granules of polyhydroxybutyrate here. But then when you knock out nitrogen fixation, that frees up all this carbon, and this non-fixing mutant is hoarding much larger amounts of polyhydroxybutyrate. So we'll look at the consequences of PHB diversion both to the rhizobia and to the plant. From the rhizobium's point of view, we hypothesized that these cells can, even in a good fixing strain, half of the dry weight of the cell could be PHB of the rhizobium cell. And so we hypothesized that that might be beneficial to the rhizobia when they're under starvation, perhaps in a soil where they're outcompeted by other bacteria for soil resources. And so to test that hypothesis, Will Ratcliffe, who's now a professor at Georgia Tech, took a single strain of rhizobia, grew them under different conditions, where they accumulated either a lot of PHB or very little, and then he starved them in low carbon soil, basically. And the low PHB cells all died out within 5 months, whereas the high PHB cells, even though they were the same strain, they were actually able to reproduce without an external carbon source, just using the PHB in their cells. And 5 years later, there were almost as many alive as there had been at when the starvation started. So he looked into that a little more deeply. And what we're seeing here on the right is two images of a dividing rhizobium cell. And so the image here on the left is GFP fluorescence. And so you can see the mother cell here is dark because the mother cell has gone dormant. It's not making protein, so you don't get any GFP. The daughter cell, though, is metabolically active and it is making GFP and presumably other proteins. But if we look over here at a PHB stain, we see that the dormant mother cell has actually kept almost all of the PHB and given very little. So the daughter cell is set out to seek her fortune without much in the way of resources. She's metabolically active. If she finds some food, things great, she can reproduce. But the mother cell is dormant, so not using very much PHB and has a high endowment of PHB. My more recent grad student, Catherine Mueller, now a postdoc in Lauren Quarter's lab at Cornell, hypothesized, well, she actually modeled this in a paper that we published earlier this year, that combination of dormancy and high PHB endowment could actually allow a rhizobium cell to survive in the soil for years without any external carbon source, assuming that starvation is the only risk that it faces. You know, if it's hiding in a crevice or something and not being eaten by protozoa. And so one of the things she did to test that hypothesis was took advantage of this long-term experiment at Waseca, Minnesota, where they have, among other things, plots that had soybeans 30 years ago, but since then have been in continuous corn, so no host for the rhizobia, and also intermediate numbers of years, the 0 here being a plot that just had soybean just within the last few months. And so what Catherine did was to collect soil from each of these plots and then do a common garden experiment where she took that soil into the laboratory and measured the rhizobia from these different soils. How much PHB do they accumulate when they're in nodules? And so what she found is that as you went to soil that hadn't had rhizobia for 30 years or even for 5 years, those soils were increasingly dominated by strains that are much greedier in terms of hoarding PHB when they're in symbiosis, actually hoarding twice as much under the same conditions as the strains that died out earlier without a host. So I really hope she'll publish these data soon. I just sent her an email about it. Well, so it looks like diverting PHB is beneficial to the rhizobia, at least in one sense, but there could be a cost to the plant because they don't have as much, they're not using as much carbon to fix nitrogen. And that was actually demonstrated quite a while ago by Ceballos, who actually made a PHB knockout of bean rhizobia. And over the course of bean growth, he measured nitrogen contribution from these rhizobia and found that the wild type strain, which was diverting energy to PHB, didn't provide the plant with as much nitrogen as his PHB knockout. Now, he kindly provided us with those strains, and we've done some follow-up work with those. And one of the questions that Catherine asked is there actually an effect of this diversion on the efficiency of nitrogen fixation? That is, on the amount of nitrogen that the plant, that the rhizobia fix relative to their respiration cost. And so she set up this cuvette that could let us do gas exchange. We're measuring CO2 from root and nodule respiration. And hydrogen, which I mentioned, is a byproduct of nitrogen fixation. Over here, we're manipulating, these figures have been published, manipulating the nodule interior respiration rate, basically, and then plotting hydrogen, which is a proxy for nitrogen fixation, as I said, as a function of respiration. And so the slope of that line is basically the incremental increase in nitrogen fix, or the marginal increase in nitrogen fixation after we do some corrections, with a marginal increase in respiration, which I would call the efficiency of nitrogen fixation, or one measure of that efficiency. And so she measured that and found early in the season, no difference, consistent with Cevallos not finding a difference in nitrogen fixation early in the season. But later in the season, the PHB knockout was apparently using some of that energy that could have gone to PHB for nitrogen fixation, and that resulted in higher efficiency on a respiration basis. Now, of course, respiration is just one of the carbon costs of nitrogen fixation. Another cost is the one we saw earlier, the cost of actually making nodules. But Ryoko Ono, who was previously a student in my lab and is now on the faculty at Santa Barbara, used those same strains and found again that the PHB negative strain produces more shoot weight relative to nodule weight. So more efficient by that criterion as well. Well, so there's a direct benefit to the rhizobia from having more PHB, but the more energy they divert into PHB, the less nitrogen they fix, and the less nitrogen they fix, the less healthy the plant's going to be, the less the plant will be able to photosynthesize and have carbon to share with the rhizobia. So given that trade-off between these two effects of diverting carbon to PHB, what should rhizobia do if they only care about their own fitness and not about the fitness of the plant, which they do. Stuart West, now a professor at Oxford, not really thanks to the sabbatical he did with me, I'm afraid, but appreciated having him visit the lab. He modeled this, and this vertical axis here is the fitness optimizing allocation of rhizobial resources to nitrogen fixation. So we could say 50%, they're putting half of their energy into nitrogen fixation and half into PHB, something like that. And he modeled that as an effect of the relatedness among strains on a plant. And so lots of people have modeled this situation or similar symbioses with the assumption that you only have a one-to-one interaction between host and symbiont. And so that's the situation over here, one clone of rhizobia per plant, therefore relatedness of 1, or 100% if you like. And under those conditions, if there's some nitrogen in the soil, the rhizobia still should put some energy into nitrogen fixation, maybe 25%, because otherwise their plant might die. If there's less nitrogen available in the soil, they should put more energy into fixation to maximize their own fitness, maybe 50% or so. But this is all with one strain per plant. If you go to 2 strains per plant, the optimum allocation of nitrogen fixation drops to 0 in a medium nitrogen soil. And if you go to realistic numbers of rhizobia per plant, like 10 different strains, that's down here, the model says they shouldn't fix any nitrogen at all. Now, this is I should point out that this situation of one strain per plant is what you usually see in research studying nitrogen fixation in the laboratory, but you never see it in the field. And the field is like this. So, we looked at this and said, well, this can't be right, because this model basically predicts no benefit. The rhizobia stop fixing nitrogen because those that do fix nitrogen get outcompeted by strains that don't. Rhizobia stop fixing nitrogen, therefore the plants are going to abandon symbiosis. The whole thing should have fallen apart in, I don't know, 100 years or so, and yet we know the symbiosis is tens of millions of years old. So obviously the model was wrong. And the question is, what was wrong with the model? Well, one possibility that we eventually looked at is work done in Europe showing that some legume species actually have the ability to manipulate rhizobia in their nodules in ways that cause the bacteroids, the nitrogen-fixing cells of the rhizobia, to swell up. And this has two effects. It greatly reduces or eliminates the ability of those cells to reproduce, but it was hypothesized to actually increase their efficiency. At least we hypothesized that. So let me show you first what this is. This would be what the rhizobia in a soybean nodule would look like. They look pretty much just like bacteria out in the soil. These are completely able to reproduce. They're viable in culture and so on. But in an alfalfa nodule or a pea nodule or a peanut nodule, a majority of the cells and all of the ones that are actually fixing nitrogen have swollen up like this. And there's only maybe a third or so, you can see that here, a third of the cells are still viable. A third or so of the cells haven't differentiated yet. They're still viable to reproduce, but they're not fixing any nitrogen. So the question is, what effect did this have on efficiency? And so Ryoko used the same kind of analysis that I showed earlier, and a couple of cases where the same rhizobium strain can either go into a manipulating host like pea or a non-manipulating host like bean. And you can see that when it was manipulated, that strain is more efficient. Same thing with cowpea and peanut, a different rhizobium strain that can go either into cowpea or in peanut, and it was more than twice as efficient at fixing nitrogen when it was in peanut. So this certainly looks like something that would be beneficial to the legume to be able to manipulate the rhizobia this way. And so Ryoko looked at this in an evolutionary context. Here's a evolutionary tree, not all the species that she looked at, actually. She looked at something like 40 of them. But she asked the question, the ancestors of the legumes, did they have the ability to manipulate rhizobia this way in their nodules? And if not, how many times has this trait evolved? And so based on her phylogenetic analysis, using Bayesian analysis and so on, she was able to show that the ancestor probably did not have this trait, but that it's evolved at least 4 or 5 times in some lupins or in baptisia, in peanuts, in medicago and pea, that may be a single event leading to both of those species. But you don't see it in phaseolus bean or in glycemax soybean. So this is something people have thought about a little bit. Would it be possible to genetically engineer soybean in order to get this increased efficiency out of the rhizobia? But going back to the model, it would be nice if they could manipulate the rhizobia, but is there anything else that they can do? And the key assumption that we made here at the left was the assumption that the only benefit to the rhizobia from fixing nitrogen was the benefit that they all share of having a healthy plant. And so we said, what if in addition to that benefit, and you don't really want to share benefits with another strain on your plant because that strain is your most likely future competitor for a new host, right? You want them to die. But what other benefits could there be? And one benefit could be if legumes somehow have the ability to monitor each nodule, and if the nodule is not fixing enough nitrogen, shut it down in a way that keeps the plant from wasting resources on that nodule, but that might also have a negative effect on the fitness of the rhizobia inside, on their ability to reproduce or survive. And so when he modeled that hypothesis, which we call the sanctions hypothesis, regardless of the relatedness and regardless of two different versions of how sanctions work, the model predicted that yes, you would get at least 50% or more of the energy that rhizobia get, they should put into nitrogen fixation in order to maximize their own fitness, even though they don't care about the fitness of the plant, except indirectly. But is this true? Well, Ryoko Ono also looked at that, and she looked at it specifically in alfalfa and pea. Both of these are species where they have the manipulation. So do they still need these sanctions in addition to being able to manipulate the rhizobia? And she found that they did. So here are alfalfa nodules in air, and in air where they are able to fix nitrogen, the nodules grow, the rhizobia reproduce inside. Some of them are non-reproductive, but there's still, a million or so reproductive rhizobia in each of these nodules. Whereas when she prevented nitrogen fixation by putting half of the root system in a nitrogen-free argon atmosphere, growth stopped. And as a result, if you look at rhizobia per nodule, the vertical axis here, which is different scale for the different species, the rhizobia that were prevented from fixing nitrogen only had a third or so of the reproduction in nodules as those that were fixing nitrogen. It was true in alfalfa and pea, even though both of those species were manipulating the rhizobia. And earlier, Toby Kears had shown that it was true in soybean, where they don't manipulate the rhizobia other than shutting down non-fixing nodules. Now, all of those data were for non-fixing nodules, but Toby, who's now a professor at the Free University of Amsterdam, a bit older than us now, I think, though she made professor quite young. She asked, okay, but what about rhizobia, like those that we saw in my second or third slide? What about rhizobia that fix some nitrogen, but not nearly as much as the good strains? And so now she made different mixes of gas. And so, for example, if you give a root system 4% nitrogen, it doesn't take much nitrogen for them to be able to fix nitrogen. But if you give them 4% nitrogen rather than 80%, then you get 50% of maximum nitrogen fixation. And then with lower values down eventually to zero, here you get no fixation at all. So consistent with her earlier results, if the rhizobia didn't fix any nitrogen or very little, then they reproduced maybe a third or a quarter as much as if they were fixing nitrogen. But these guys here, potentially diverting half of their energy into PHB or otherwise into their own reproduction, the plant didn't always catch what they were doing and only shut down maybe one nodule in 6, something like that. So it looks like, at least with the one soybean cultivar that she studied, that rhizobia could get away with diverting half of the energy they get from the plant into their own selfish uses, and not usually get hit with sanctions. If they divert more than that, they're in trouble. And I think this is probably why most rhizobia out there are kind of mediocre. They can get away with diverting a fair amount of resources without getting hit with sanctions. Now, the diversion is not the only reason something might be defective. I mean, these guys are just defective, right? They don't know how to nodulate. So Toby Kears just did that work with one strain. And so my main focus now, I'm looking at a dozen strains that I got from our soybean breeder, Aaron Lorenz. And that's one of the things I'm going to be looking at is, do they differ in how they treat mediocre rhizobia? They're fixing some nitrogen, but not very much. Now, so that's something, if I find something, I hope that can go into our soybean breeding program here. But that would be, one way perhaps to get at this would be some kind of genetic engineering of the plant to change how they deal with mediocre. Whoops, I didn't want to do that. to change how they deal with mediocre rhizobia. And we know a little bit about the mechanism, though not as much as I would like. My grants to figure this out have all been rejected so far. But here's the method that we used. So it turns out that legume nodules, including soybean nodules, have a hemoglobin. And like the hemoglobin in our blood, that hemoglobin changes its light absorbance when it binds oxygen. So this is just like the device that they used last week to find out that Donald Trump had low blood oxygen. The difference is that we can do some things with root nodules that we can't do with Donald Trump. For example, here, we have nodules in air, and then we switch them to a oxygen-free nitrogen atmosphere, which we couldn't actually do with Donald Trump, but it's very useful because it tells us where zero oxygen is, because that's where it levels off in the absence of oxygen. Then we switch to a high oxygen atmosphere, 70% or so, and in 1, 2, 3, 4, 5 seconds, the hemoglobin saturates. So now we know what 100% saturation is. It's that level there, and that's where this scale came from. Now, you may have learned in a class or in a book that the hemoglobin in legume nodules protects them from oxygen, and that's totally true. It can protect them for 5 seconds. The real protection long-term is respiration and a physical barrier to gas diffusion surrounding the central zone. And we have known that since 1983, a paper by Appleby. So please don't tell your students that leg hemoglobin protects nitrogenase from oxygen. So this is before grazing. After the sheep went in and grazed, here's what we found. Basically no oxygen in the nodule. Now that could have come from a huge boost in respiration, unlikely in nodules with no leaves, but we know it's not true because the slope of this line here is the respiration rate, and it's no different than it was before. What's different is how fast oxygen diffuses into the nodule. It now takes forever because the oxygen gas permeability is so low. And we can actually calculate the oxygen gas permeability from this kind of assay when we get good data. Sometimes it's too noisy. And so we actually did that for these sanctions in alfalfa. I'm sorry, in soybean. I think this might be alfalfa nodules, but this graph is for soybean. It's from our 2003 paper in Nature. And sure enough, the oxygen level in nodules that weren't fixing nitrogen goes down to half the level of what it is in the control nodule. So it does seem like possibly the mechanism of sanctions involves control of nodule gas permeability. The mechanism of that, we think, am I really at about an hour? I'm almost done, but the mechanism of that, we have some evidence from my former colleague, Tom Kinraid, who unfortunately passed away last month, but he was an electrophysiologist, and we have these data from 1995 suggesting a mechanism that's roughly analogous to what happens in guard cells, potassium channels opening and changing turgor of cells, and that changes the gas pathways into the nodule. Could somebody tell me whether I really only have 2 minutes left? That's what my clock is saying.
No, you can keep going.
Okay.
We usually wrap up around 4, but we like to have a few minutes for questions.
Yeah, I'm almost done.
So plenty of time still.
So this is my scheme for selecting soybeans that would have stricter sanctions and perhaps thereby benefit future soybeans growing in the same field. So this is in my book, so you can look it up. But you start out with a diverse population of legume genotypes, measure the yield, but don't select from this population based only on that yield. Instead, you go into the same soil with a genetically uniform test crop of maize. These are all the same maize genotype, but these two plants grew more because of some effect of this soybean, these two soybean genotypes, perhaps an effect on mycorrhizae, like Nancy Johnson showed many years ago here in Minnesota. And then you go into a single genotype of soybean as a test. And here, this one, in addition to growing better in the corn year, is growing better in the soybean year, perhaps because of stricter sanctions imposed by this soybean back here, that it only let the very best local rhizobia reproduce in its nodule, release those into the soil in numbers that swamp soil populations. And therefore, 2 years later, given the 30-year survival that Catherine Mueller showed many slides ago, 2 years later, we see that benefit. That's the hypothesis anyway. So this is just a summary of stuff I've already said. Soybean strains differ in net benefits, partly due to differences in resource diversion. Some legume species can manipulate rhizobia, but perhaps more important than that is differences among legume cultivars in their ability to impose sanctions, where they shut down at least non-fixing nodules, and maybe we can find a soybean cultivar that shuts down mediocre nodules and only lets the very best ones flourish and become available to future generations. So I will stop my, oh, don't forget to buy my book, and I will stop sharing and take questions.
Thank you for any questions for Dr. Denison?
So if the leg hemoglobin isn't there to, or isn't doing a very good job of protecting the nodules against oxygen, what is it there for? Is it just adding some benefit to the oxygen protection, or?
No, really nothing to do with that. Good question. It does the same thing that does on our blood. It transports oxygen. And so the problem that legume nodules face is they have to have a very low permeability shell around the interior to protect the interior from oxygen. You have this low permeability shell, you've got respiration inside, and the result of that, going way back to John Tejepkema, who died a couple of years ago, is that the oxygen drops going into the nodule interior, drops to basically undetectable levels, undetectable by electrodes. Well, that's great for protecting nitrogenase, but it means you don't have any gradient to drive a diffusion of oxygen to where it's needed by the rhizobia respiration. So the oxygen can diffuse through the nodule, we think, through air-filled pathways, and diffusion is pretty fast in air, even with a low gradient. But then as soon as they get to the surface of an infected cell, they have to diffuse through liquid, which is super slow, and they've got no gradient driving that, no difference in constant. It's like Ohm's law, right? No difference in concentration driving their diffusion into the rhizobia where it's needed. But they do have a steep gradient, not of oxygen, but of oxygenated like hemoglobin. So even though the hemoglobin molecule doesn't diffuse as fast as an oxygen molecule because it's bigger, that super steep gradient from near 100 at the surface to 0 in the interior, that's what drives the diffusion. So that's the main thing it's doing. It probably provides some kind of transient protection, but that's the main thing. And as I say, there was a review by Appleby back in '83 that pointed this out, but it somehow, some people have forgotten it, apparently. I would add that, you know, there are efforts underway to get wheat or something to fix nitrogen. And they all seem to have the idea that if you put the nitrogen fixation genes in, everything will be fine. But this oxygen problem is really, really difficult. because the only way to get enough oxygen, the only way to get enough energy to fix a decent amount of nitrogen is respiration. You can't have respiration without oxygen. And yet, if you go to 0.5% oxygen, you fry your nitrogenase. So you have to have two things at the same time. You have to have a very high flux, a very high movement of oxygen into the nodule, but it has to be moving very fast at very, very low concentration. Not only that, what happens at night? Let's say you've got this nodule and it's got enough respiration to consume the oxygen that goes, into the nodule, and then it's nighttime and the temperature goes up and the respiration drops at lower temperature. A 10% drop in respiration would fry nitrogenase if the gas permeability stayed constant. So they have to have this barrier, and not only that, they have to have the barrier, and the barrier has to be under physiological control so that when the oxygen starts to rise inside, they can shut down that barrier to keep the oxygen at a non-dangerous level. And that's what I showed in that picture of the grazing study. What you would think, sheep eat the leaves, no more photosynthate, nothing to power respiration in the nodule. Without respiration, the oxygen goes up and fries nitrogenase. But exactly the opposite happened because they shut down that nodule so tight that no oxygen could go in. So they're not respiring in there. They're not using any of the energy that's in that nodule. And as soon as the photosynthate comes back, the leaves regrow, they open it up, and they're back in business. The nitrogenase is still functional. Long answer, but.
That was helpful. Thank you.
Ford, you were presenting some results showing how some strains hoard for more PHB, and those are able to survive in the soil for more years.
Yes.
I was wondering, in some agricultural systems, we do rotations that sometimes can be for a number of years. I was wondering if you think that that system is indirectly selecting for strains that are taking more resources and hoarding more, because those are more have more chances of survival.
That's a very good question.
The size of the rotations in agricultural systems will not have a significant impact. That's a very
good question, and I'm tempted to give a one-word answer, which is yes. But actually, my student Catherine is trying to model that situation at the moment, and that's why we haven't published those data yet. And she's going to try to combine it with a model. She actually found some kind of strange results, which, let me see if I can get this right. The simplest hypotheses that she could come up with would either explain what happened between 1 year of corn versus 2 years of corn versus 5 years of corn, or what would happen between 1 year of soybean versus 5 years of continuous soybean. But she's had difficulty. I think she's got a hypothesis that can explain both of those results, but it's not a simple hypothesis. And we always like simple hypotheses if we can find them. What she came up with involves a lot of frequency dependent responses, which are not impossible, but not as simple as you might like. But your thinking is absolutely on target. If all else being equal, if things were simple, yes, going out of soybean for a longer period would select for not just PHB hoarding, which would directly select against benefits to the plant, but also the longer they're in the soil, if there's any kind of trade-off between survival in the soil and getting the plant to give them resources in a nodule, any kind of trade-off like that, you would expect to get worse with years between rotations. But it may be more complicated than that, but you're definitely thinking the right way. I know this is a lot. If people haven't thought about this stuff before, it was a lot of material to avoid, to absorb, I hope. If some fraction of you find it interesting enough to look up some of our papers, or, the great thing about YouTube is you can actually pause when I'm talking too fast and think about what I said. I'm doing that quite a bit with seminars these days, which turns an hour seminar into an hour and a half seminar because of the, I'm stopping to look up one of their papers or something like that. But anyway, that is an option.
No, this was great. Thank you. I do have another question.
Okay.
I found very interesting what the slide that you presented towards the end, where you show the rotation of soybean and corn and soybean, and where you found in the third generation what was one of the cultivars initially that provided a benefit. How do you think that can be measured in yield, because the relative benefit might be relatively small? So that should be coupled with other types of measurements, right? Or do you think?
Yes, no, you're right. So all of this work I'm doing, first of all, you understand that was a proposed experiment, not something I've actually done.
Yes.
And you may remember I tried to get people at the soybean breeders meeting to do that experiment. But all of the work that I've talked about, hello? Something just happened. I don't know if people are still hearing me or not. I just saw an iPhone pop up. Okay. All of the work that I've talked about, I'm pretty confident, 80 or 90% confident that it's correct in principle, that these effects really are there, that there really is a trade-off between fixing nitrogen and longer survival in the soil, and so on. What I'm not at all sure of is, and I expect there'll be differences among cultivars in this, what I'm not at all sure of is whether the differences among cultivars will be large enough to make it, worthwhile following up on this in real field experiments with adequate replication and, 2 years of data and so on. And even if it turns out that there are large effects, large differences among cultivars and in their release of good versus mediocre rhizobia into the soil, is that, we're pretty sure that some rhizobia can survive for a couple of years to the next rotation, but is that effect going to be large enough relative to all the other, things that determine rhizobia survival in soil? Because, the first year you do this, there's probably going to be at least a little bit of a yield hit, because the plants are shutting down some nodules, they're shutting down mediocre nodules, and maybe they don't have enough time to make new nodules or replace them with better strains and so on. So there could be a bit of a first year yield hit, and will that be enough? But the thing I'd really like you to take from that graph isn't just nitrogen fixation, isn't just mycorrhizal fungi, isn't just microbes, but the theme of my book is that the opportunities to improve crops are the things that natural selection didn't already do. So it's not, trade-off free drought tolerance. If drought tolerance was trade-off free, natural selection already did it. It's the things where there were trade-offs that prevented something from evolving naturally, but where it's a trade-off that doesn't hurt us agronomically in modern agriculture. And so one of those trade-offs is, from the standpoint of the fitness of a plant, your neighbors are your enemies. They're your competitors for resources. Their seedlings are your seedlings competitors. You want them to die. Whereas in agriculture, we've got a single genotype. They should all keep their roots in their own space and not waste resources on making roots to invade their neighbor's space because their neighbor is another, another Chippewa or whatever the soybean cultivar is. So one of the things that natural selection never does is invest very much in future generations. Now, a plant will invest in its own seedlings by putting nitrogen and starch into its seeds, but it's not going to invest in the collective benefits to its seedlings and their competitors by only releasing good rhizobia into the soil, because yeah, that would benefit its seedlings, but it's mostly going to benefit its competitors because that's, it's this mixed situation. So natural selection is not going to favor traits that benefit future plants of the same species growing in that field. But both in terms of effects on microbes, you think about things like persistent root channels. If we had a plant that its root would decay, living this kind of, nice loose network that future roots could go down and get into the soil cheaply, not have to break through a hard pan. Natural selection wouldn't give you that because it would be benefiting everybody, not just that plant's own descendants. So that's really what I'm saying is when we think about plant breeding, we think a little bit about how this year's species affects next year's crop growth. But let's think a little bit about how this year's genotype, this year's variety affects next year's crop growth and the year after that. That's the main point of that slide. And rhizobia would be one example, but not the only one.
Can I ask a question?
Please.
I post my question in the chat box. I don't know if you can access to it or not.
Oh, yes, I will open chat. I'm
The long
sorry.
question.
Would you tell us effective grazing, that one?
No, the next one.
We should not tell students what like hemoglobins protect nitrogenase. Yes, we should not tell them that. That's wrong.
Okay. There's another question I also post. I don't know if you can see that or not.
That's the last one I'm seeing so far is the one about hemoglobin.
Okay, maybe I can ask you directly.
Yes.
So maybe I just read it. Ecologist and geneticist.
Oh, I'm sorry. I do see that just earlier.
Okay.
Same biological issues, different methods, terms, reconcile, cheater and sanction concepts. Okay. My evolutionary biologist colleagues argue a lot about these terms. So they may not be the greatest terms. Let's see. Between sanctions and incompatibility, okay. So basically the way I would define cheating, cheater is a difficult concept. Maybe we'll get into that, but the way I would define cheating is doing something that potentially benefits yourself at the expense of your partner. Okay, so and really it's doing that more than others of your species do. So really if rhizobia divert carbon to PHB, who are they cheating? Well, maybe they're cheating the plant, but how much nitrogen should they fix? I mean, it's, we don't have morality here. It's really hard to say that fixing a certain amount is cheating and fixing more is not. That doesn't really make sense to me. But if you're a rhizobium and you're not doing your part, you're not fixing your share to keep your shared host alive. Just like if you're not if you're not contributing to public radio, if you're not doing your share to keep public radio on the air, those are the cheaters. So really what they're cheating is the other rhizobia on that same plant. Your issue about incompatibility would show up that something that's incompatible or defective would not benefit. It would be hurt. The rhizobia would be hurt. Cheating benefits the rhizobia, but hurts the plant. The problem is that really cheating would benefit the rhizobia if you had a hypothetical legume without sanctions. Okay, given plant sanctions, it makes it trickier to figure out whether something is cheating or not. But I would say that, for specific examples, if you're diverting more energy to PHB than the other strains on the plant, you're cheating. Incidentally, what about, can I take another couple of minutes?
Yep.
What about these?
Yes. You
can leave if you need to. This situation where the legumes manipulate the rhizobia. So here you've got nodules, they're kind of thumb-shaped. You've got these nodules, and 1/3 of the rhizobia in that nodule are still reproductively viable because they haven't become bacteroids yet. 2/3 of them, the ones that are fixing nitrogen, can't reproduce. So there's no point in those rhizobia, those bacteroids that are fixing nitrogen, there's no point in them hoarding PHB because they're not going to have any descendants. So why do those plants need sanctions if cheating doesn't work for the rhizobia anyway? Well, it turns out there's another way. Because in those symbioses, and only in those symbioses, that is not in soybean, for example, some fraction of those rhizobia strains have a trait where the nitrogen-fixing cells, the bacteroids, can take carbon that they get from the plant and turn it into this molecule that the plant can't use, which can then diffuse along the nodule to their reproductive clone mates. And those reproductive clone mates can take that up and use it for their own, reproduction and survival. So it's a different way of cheating, of diverting resources. And again, I've, submitted grant proposals to explore that, and so far they haven't been funded. But my guess is those are not the only ones. I mean, one other possible way is this hydrogen business, because we know that some strains of rhizobia make more hydrogen and therefore fix less nitrogen than others. And we know that rhizobia can use hydrogen as an energy source. So it's possible that making hydrogen, some of it seems to be inevitable, but making hydrogen may actually be another cheating mechanism. But that's speculation.
Thank you.
That's an awful lot to absorb. I'm happy to take more questions, but feel free to go on with your day. I had to think about the effect of grazing on nodules and nitrogen fixation. Yeah, basically it shuts it down. And the assumption back when I was a grad student is that grazing shuts down nitrogen fixation because they don't have any photosynthate. And one day when I was going for a walk towards the end of my PhD, I said, wait a minute, that can't be right. Because if they don't have enough photosynthate to consume all the oxygen that goes into the nodule, they lose nitrogenase, it's fried. The nodules can't be limited by photosynthate directly. They've got to be limited by oxygen. The oxygen permeability has to be low enough that whatever respiration they've got left after grazing, that's enough to consume all that oxygen, because if they don't, they lose nitrogenase. And it turns out that a Swiss scientist, Uli Hartwig, actually did an experiment with clover where he defoliated clover, whether it was grazing or mowing, I don't remember. And then he raised the oxygen around the nodule. And sure enough, if you raise the oxygen, you can restore the nitrogen fixation. And temporarily, I mean, you could run out of photosynthate. So basically, after grazing, the activity is really limited by oxygen permeability. And if it weren't, they would lose nitrogenase.
Thank you. Are there any final questions? Well, thank you very much. We really enjoyed the talk and appreciate your time to be the guest speaker.
I really appreciate the opportunity to talk about my specialty for a change. I did that some at the soybean breeders meeting, but a lot of my talks, for some reason, I get asked to talk about other things. So this has been fun.
It was very interesting. Thank you.
All right.
Okay.
Bye.